Welcome back to Digital Teacher YouTube channel. Still on this series of genetics. In our last video, we did explain that when Mendel crossed purple flowered pea plants with white flowered pea plants and planted the seeds from this cross, he produced plants with purple flower in the F1 generation with no white flower plant at all. However, when he crossed two plants from this F1 generation, he produced two kinds of plants with some having purple flower and few having white flower in the ratio 3:1. So we named the plants gotten from the first cross of Mendel the F1 generation, also called the first filial generation. So Mendel named this color of flower a character. Did Mendel work on other characters of pea plants? Yes, he did. He worked with characters like the plant height, the flower position, the seed shape, the seed color, pod color, and all other characters. And he obtained a similar result like his first cross. For example, when he crossed plants with round seed with wrinkled seed in the F1 generation, he produced plants which all have round seed, having resemblance to only one of the plants and no resemblance with the other plants having wrinkled seed. Similar result was gotten for other crosses. So crossing a plant with purple flower with white flower produced in the F1 generation all round purple flowered plants. But in the F2 generation, two sets of plants were produced. Mendel concluded that when plants with opposite characters are crossed, one character is always expressed, while the other one is not expressed. This one that is expressed is called dominant trait, and the one that is not being expressed in the F1 generation is called recessive traits. So looking at the seed shape, the round seed trait is dominant over the wrinkled trait. So when it crossed pea plants with round seed with pea plants with wrinkled seed, in the F1 generation, he obtained plants which are all having round seed, showing that the round seed is dominant over the wrinkled seed. Similarly, when he crossed plants with yellow seeds with plants having green seeds, in the F1 generation, he obtained plants which are all having round seed. Similar results was gotten with the color of pod, the shape of pod, the height of plants, the flower position, and the flower color we studied earlier. So the question is, what has happened to the recessive traits in the F1 generation? Could it be that these traits has disappeared from the line? No, we'll look at that in the next cross. We now know that the purple color was expressed because it carries the dominant gene, while the white color was not expressed because it is controlled by a recessive gene. Let's understand this by looking at the procedure. If P is used to represent the allele or factor that produce purple flower, and P in lowercase is used to represent the allele which produce white color flower. Any idea why we have to represent this character with double letters? This is because it is a diploid organism. What do we mean by diploid? Let's get to understand this term. It is known that in humans there are 23 pairs of chromosomes, right? A member of each pair from the father, while the other is inherited from the mother. So when an organism has two sets of chromosomes, it is described as diploid. Let's get back to our cross with purple flower and white flower. So, representing the allele for purple flower with double capital letter P and the allele for white flower with double small letter p. This will separate and give us the allele capital letter P and allele capital letter P for purple color. This will also split to give us the allele small letter p and small letter p representing white color of flower. So let's do the crossing. We'll cross the p from one side with the p from the other side. So we obtain a capital letter P and a small letter p. Similarly, when we cross this other p again with the second small letter p from the white color, we'll get the capital letter P and small letter p. We do the same for the second letter or allele, and let's see what we get. So we can see that all the plants are having both the allele for purple flower and the allele for white flower, but they all are having purple color in the F1 generation. So let's cross the two plants from this F1 generation and see what the result will be. So the allele are separated now. Let's do the crossing and see what we will get. When we cross the first one, we'll get double capital letter P representing purple flower. We do the second one also, we get a capital letter P and small letter p. So let's cross the allele for white flower with the allele for purple flower. And that's what we get. Lastly, the two allele for white flower combine to give us double allele for white flower. So these results produce two different kinds of plants in the ratio 3:1. That is 3 purple flower and 1 white flower. The purple flower can be expressed in homozygous form or heterozygous form, while the white color is only expressed in homozygous form. What do we mean by these two terms, homozygous and heterozygous? Let's get to understand that. Homo means same, while hetero means different. So homozygous means same set of genes, that is, both are dominant or both are recessive. This is seen in the case of double capital letter P and double small letter p. So what does heterozygous means? It means different set of genes. This is found in the case of capital letter P and the small letter p allele. So one dominant and one recessive. So the dominant allele is always expressed in both homozygous and heterozygous form, but the recessive allele are only expressed in homozygous form. Let us understand this with another cross. Let's say the character, height, and the traits are tall and dwarf. So we are crossing a tall plant with a dwarf plant. So we are going to represent our height with letter T. So since tallness is dominant over dwarfness, we are going to represent the tall character with double capital letter T and the dwarf character with double small letter t. So this is the parent plant. Let's cross it and see what we will get. Just like in our first example, this allele will separate to give us a separate allele, and this also will separate to give us separate allele. So when we cross the allele for tallness in the first plant with the allele for shortness or dwarfness, we'll get heterozygous tall. Similarly, we cross the tall allele again with the dwarf allele. We are going to obtain heterozygous tall. We do the same for the second allele for tallness. So as we can see, all the F1 generation plants are having heterozygous tall. They are all tall. So let's cross two plants from this F1 generation as we did in our first example and see if we obtain a similar result. So we cross this. After they separate into their individual allele, let's now cross it and see what we will get. So when we cross the allele for tallness with another allele for tallness, we obtain homozygous tall. And when we cross the allele for tallness with that of shortness, we obtain heterozygous tall. We cross the allele for shortness with tallness, we obtain heterozygous tall, and lastly the allele for shortness combined to give us homozygous dwarf. So in the F2 generation, we have obtained also two sets of plants, 3 tall and 1 dwarf plant, ratio 3:1. So 3 plants are tall, while 1 is a dwarf plant. With this result, let's get to know terms in genetics. The two terms are phenotypic ratio and genotypic ratio. The phenotypic ratio is 3:1. That is 3 tall and 1 dwarf. While the genotypic ratio is 1:2 to 2:1. That is 1 homozygous tall, 2 heterozygous tall, and 1 homozygous dwarf. What does the term genotypic ratio and phenotypic ratio means? Let us understand this with the following illustration. The features that can be seen or observed by us externally are called phenotype. They are observable characteristics. So in the case of this F2 generation, what we observe is 3 tall and 1 dwarf. So that is why the phenotypic ratio is 3:1. This is because the gene for tall height could either be in form of homozygous or heterozygous. So a plant having two alleles for tallness and a plant having an allele for tallness and an allele for dwarfness will both look tall. Genotype, on the other hand, means genetic makeup that cannot be observed. So physical expression of the gene giving rise to that particular observable trait is called phenotype, while the genetic makeup that cannot be observed is called genotype. So it is the set of genes that give rise to a particular trait. It is called genetic makeup. So the tall height is the phenotype, while homozygous tall and heterozygous tall are the genotype of the height. Similarly, for hair color, black color hair is the phenotype, while homozygous black or heterozygous black would be the genotype of the color. Back to our illustration of stem height. The genotypic ratio is 1:2:1. That is 1 homozygous tall, 2 heterozygous tall, and 1 homozygous dwarf. While the phenotypic ratio is 3:1, that is 3 tall and 1 dwarf. So in summary, a diploid organism is an organism having two sets of genes. Homozygous means both the alleles are same. Heterozygous means both friends. Dominant means the genes which are always expressed. Recessive means genes which are usually suppressed and expressed only in homozygous form. Phenotype means externally observed character, while genotype means the genetic makeup of an organism. Did Mendel only study one character at a time? No, he studied 2 and 3 and 4 characters at a go. Let's get to know more about this in our next video on dihybrid cross. Till I come your way next time, keep on reading.